In this video, we're going to take a look at some different ways that we can use to control microbial growth. And this goes back to this idea. If you look at this figure here, you can see that the interior of your car has got some really dirty places. And if we look at the average number of colony-forming units, or remember a CFU is a single cell, you can see that there's a wide variety of different numbers of cells that were found when they measured it looking at this, and there's some dirty places out there. If you look at the center console or the inside of the steering wheel right there, places where you're going to have your hands, maybe you're dropping food or drink right there, those tend to be really rich in the number of microbes that are hanging out on that area. And so we would want to be able to have some way of getting rid or decreasing the number of these cells that are out there to try and decrease especially the transmission of things like pathogens that are out there. And if you look at this little guy, you can see that the Center for Disease Control actually does rate the different kinds of microbes out there into what are known as biosafety levels. And the BSLs are basically used to label labs out there so you know what level of danger you can be encountering. And the stuff that we're using in lab is considered biosafety level 1 right here, because they don't cause disease in healthy hosts, or they pose a minimal risk. And so when we look at this, these BSL levels are really rated on the danger they pose to laboratory personnel, but also to the community in general. All of us already start out with E. coli, and if you work your way up, you're going to run into different forms of these microbes that can cause diseases. So if you had a pathogenic strain of E. coli, that thing over here could be associated with a disease, and it would pose a moderate risk. So it could potentially cause diseases in the laboratory personnel, or if it got out, it could cause problems in the community there. When you go up to biosafety level 3 and 4, now you're running into the ones that could potentially cause lethal diseases, especially things that we worry about like aerosols that would be caused by pipetting or transferring things that are out there. Remember that sizzle sound I always complain about people making when they're doing liquid cultures? That's making an aerosol, and now your little microbes are traveling around in these small droplets that are produced when you have that sizzling sound, and then you could inhale those and get some form of a respiratory disease as a result. So we aren't going to be running into these BSL levels, especially the higher ones at 3 and 4, because those things you're really restricted in the laboratory space, but you can run into these kinds of things out there in nature. Again, think about tuberculosis, and then these Ebola virus or hemorrhagic kinds of viruses that are out there that are definitely out there on the planet, and that we can run into it, especially as we encroach on different areas on the planet where these viruses might be residing and they haven't caused diseases in humans recently. If we really consider this idea of we're looking at different ways to control the growth of the microbes, just like the growth of the microbes can be exponential or logarithmic growth, we can also look at the opposite type of curve, and what we see here is an example of a microbial death curve. And so you're looking at time along your x-axis and then the number of cells. You can look at this really as the number of surviving cells that are out there. And we use these death curves to try and measure something called the decimal reduction time, or the D value. And when we look at this, it's really the slope of this line is going to give you your D value, which tells you the amount of time that's needed to kill about 90% of the population. Because when we're looking at this stuff over here, remember we're looking at a logarithmic number over here as opposed to the raw numbers. And so we can see this D value is giving this our drop in the number of cells, so giving us our 90% death of the population, depending on what particular kind of protocol you're using, whether or not it's a physical method or a chemical method to try and kill off your cells. And when we're looking at this death curve, we have to consider a couple of other factors that can play a role really in the sensitivity of the cells that you're trying to examine right here. And one of the things that we can look at is the types of cells that are out there. So if If you think about this, an envelope virus is a pathogenic microbe that's out there, and these can definitely cause diseases, but because it has that membrane envelope around the outside, this virus is really sensitive. If you can pop that envelope, you get rid of the spike proteins. You don't have to worry about the virus being able to stick to the host cells, and so that can really affect the amount of pathogenicity or the ability of this microbe to cause diseases. Gram-positive cells have that thick peptidoglycan, which is very porous, so chemicals can get through there easily. And we work our way up. Our non-envelope viruses are going to be protecting that nucleic acid. And you go all the way up through these different things, and you can see gram-negative cells, because of that outer membrane, tend to be more resistant than a lot of these things that are out there. And then we work our way all the way up here. And this idea that we can look at different resistant forms of these things. So a spore for a fungus, or a cyst for a protozoan cell, or your bacterial endospores. Those are all going to affect our ability to get rid of this microbe before it can actually pose a chance of causing a disease. And you can see the most resistant form that is out there of a pathogenic particle is our prion protein. And if you remember from before, your prions are acellular proteins that fold up into this really stable shape that has these beta sheets that are going to cause that protein to not be degraded easily. It can refold itself after you denature the protein, but this is a very pathogenic protein that can cause death of cells in your nervous system. Some of the other factors we can look at, and you can see this little graph right here, that your vegetative cells are going to be killed off much easier compared to your spores that are out there. So if you think about this, this is going to cause you to have to do these treatments for a much longer time to try and get rid of those spores so they don't pose a chance of a disease in people that are out there. We can also look at the amount of time for this treatment right here, and you can see that we do have to treat these cells for a good amount of time to actually get them to the point where you drop the number of live cells after this treatment right here and hope you actually reach a point where there are no more live cells available. And likewise, if you have a higher number of cells, if you look at this, you can see your D value is going to be equivalent for these two things. The slope of both of these lines is the same, but if you start out with a higher number of microbes, it's going to require a longer time in order to reach the same levels of death. Again, when you look at that D value, you're killing off 90% of the population over that time frame, so it's going to take longer for this to actually work. One of the other things to consider associated with this time is whether or not there's organic materials that can interfere with the action of what you're trying to use to kill things off. And when we look at this, we need to consider two really important features, and that's whether or not the treatment or the chemical that you're using is going to be biostatic or biocidal. And when we look at the root of this word, static versus cidal, think about stasis is where you're actually just making something go to sleep, kind of is the equivalent there. So really, if you're stopping the growth of those cells, that doesn't mean that you're killing them off. They're just going into stasis, and maybe you kill off the cells that are actively growing in logarithmic phase, but when things go into that stationary or death phase, they can go into stasis, and if you are not metabolically active, your cells are going to be a lot less sensitive to these kinds of chemicals. When we look at something that is biocidal, or here microbiocidal, here you have the same root as something like suicide. This is actually an agent or a treatment that results in the death of the pathogen that is out there. And this is a really important distinction to think about. If you have an elderly patient or an AIDS patient and you give them a static drug, you might treat the pathogen and cause it to go to sleep, but that doesn't mean it's going away. And once you get rid of the apparent signs that it's there in the patient and you take them off the drug, now that biological pathogen is able to basically wake up and start the infection all over again. So this might seem like a minor difference unless you're a patient on the other side of the bed there who's getting treated. And what we want to consider when we look at this is these different kinds of protocols that can be used to control the growth of the microbes. And we can look at two different ways of treating things. One of them is on fomites and the other is on living tissue. And so a fomite is just an inanimate object that can be like the inside of your car, where these microbes are sticking to it and they could be transmitted to other people that are out there. And if you look at this, there are different levels of treatment that can be used. The best one that we really want to try and get as much as possible is this idea of sterilization. And when you look at sterilization, you can see you're eliminating all the cells, whether they are live vegetative cells that are actively growing or your spores that are dormant cells, and you're also getting rid of your viruses that are out there. And so when you consider this idea of sterilization, that's something that should not pose any danger of causing a disease because you've gotten rid of everything. And that isn't as easy as you would think for some of the things that are out there. NASA's been trying for decades to be able to sterilize the inside of the stuff that they're sending the astronauts up, but no matter what they do, they always go in there and they sample it and they keep finding the presence of cells. So despite their best efforts, they can't really sterilize. What they can do is disinfect and sanitize these areas. And when you're disinfecting, you're basically reducing the number of microbes that are there. And this is just reducing the microbial load. So this would be something like when you bleach your desktop, you're disinfecting the area by using your bleach right there. So you aren't necessarily getting rid of everything, but you are trying to destroy the majority of the microbes that are there. And when we look at sanitization, this is a term that's really associated with public health. And here you are again reducing your microbial load on inanimate objects. So think a sanitized toilet, or you sanitize silverware and dishes in a restaurant, and you're reducing the microbial load to the levels that the public would be affected by it. When you're dealing with patients and you're dealing with living tissue, now you've got that problem of that extra organic material being present. And so you really have to consider what's going on in terms of I can cause damage to the microbes, but I might end up causing even more damage to the skin or the tissue where I'm applying that. And when we look at this antisepsis, again, there's that root. Sepsis is contamination, so we are doing something that is against contamination to reduce your microbial load. And antisepsis is really using a disinfectant on living tissue right here. And the last one is this idea of degerming. And here again, you are reducing microbial load, but this could be something like scrubbing your hands together in water is actually degerming because you're reducing the load right there of the microbes just by going through that process. The last thing that I'll show you on this is we're going to take a look at these different kinds of control methods. And this is a figure I put on Canvas for you to take a look at because I stole it from another textbook that was out there. But you can see there's a variety of different physical agents that can be used to sterilize or to disinfect. Again, this depends on the time and the amount of treatment that you're doing. You could disinfect something, and if you expose it for even longer, that could reach points where you're disinfecting stuff. We've got our chemical agents that are out there, and again, it could be antiseptics on living tissue or disinfectants that are being used on fomites. And then the last one we can see over here is these mechanical methods that can be used to remove the microbes or decrease the loads of them. And we'll take a look at this in some other videos.